glad you came in today. Unfortunately, the test results came back and you have a mutated version of the P53 gene, making you more susceptible to certain types of cancer.
Oh no. Does that mean I have to do chemotherapy or radiation or surgery?
In some situations, yes, but luckily we have this new procedure involving CRISPR-Cas9, which is a way to change the DNA sequence in order to get rid of the P53 mutated gene and replace it with a wild type gene.
How does that happen?
CRISPR-Cas9 was originally discovered as a way bacteria kill viruses, but can actually be used to edit genes in humans. The CRISPR-Cas9 protein is made up of a cutting enzyme, or nuclease, and a DNA sequence that matches a specific sequence in the gene where we want to change. The protein will match with the gene and cut it in a very specific place. This can be used in as many cells as you want, and will give the exact same result every time because of how specific the DNA is. If another sequence is added into the cells with the correct ends, a new gene can be put where the old one was. To target cancer, the P53 gene would be edited. This gene codes for a self-destruct sequence that in healthy tissue kills cells if they start growing abnormally. If the P53 gene is corrected with the CRISPR-Cas9 system, tumor incidence could decrease.
Methods of delivery for CRISPR-Cas9. There are three main ways that we will be discussing today. One is lipid-mediated. The CRISPR-Cas9 sequence is inserted into a hollow ball of lipids, which is the same material that makes up the cell membrane. This ball of lipids is then engulfed by the cell and the gene sequence is inserted into the cell. Another method is through a process called hydrodynamic delivery. In this process, a large volume of the CRISPR-Cas9 sequence is injected and taken up by the functional cells of an organ called parenchymal cells. The gene is delivered to the parenchymal cells and incorporated after the injection process. The last method we will discuss is through viral delivery. A virus is a special type of organism that introduces their genetic material into the host cell. For CRISPR-Cas9 delivery, a virus is made that contains the genetic material of CRISPR-Cas9, and this virus is then introduced in the body to deliver the gene sequence.